See the question. What is gene therapy? Illustrate using the example of adenosine deaminase deficiency. So let us first explain what is gene therapy. Gene therapy is a technique for correcting a defective gene through gene manipulation. It involves the delivery of a normal gene into the individual to replace the defective gene. So let me illustrate this gene therapy with the example of adenosine deaminase deficiency. So this adenosine deaminase is an enzyme which is very important for the normal functioning of the immune system. The individual suffering from this disorder can be cured by the transplantation of bone marrow cells. Why we transplanting the bone marrow cell is because these adenosine deaminase enzymes are produced in the lymphocytes. In turn, lymphocytes are produced in the bone marrow cells. So here we are transplanting the bone marrow cells. So let us see this example in detail. So the first steps involved in this gene therapy is the extraction of lymphocytes from the patient's bone marrow. Second step, a functional gene for ADA is introduced into the lymphocytes with the help of retrovirus. Third step, these treated lymphocytes containing ADA gene are then introduced into the patient's bone marrow. So fourth step, so because of this reintroduction of the corrected ADA gene gets activated in the patient's bone marrow and starts to produce the functional T lymphocytes, so activating the patient's immune system. So let me explain with this figure. The first step is the extraction of the lymphocytes. So these lymphocytes are defective in the production of the adenosine deaminase. Second step, now these lymphocytes are cultured in the laboratory. So the third step is we want one retrovirus carrying the normal ADA gene. So this is the retrovirus. Here it is the genome of the retroviruses. So here, with the help of rDNA technology, we have introduced, imagine this is the ADA gene, we have introduced this normal ADA gene in the retroviruses. Now, these retroviruses carrying the normal ADA gene are infected to lymphocytes with the deficiency in the ADA production. So this is the infection with the retroviruses. So after the infection, these retroviruses transfer that ADA gene into lymphocytes. So now this is the lymphocytes infected with the retroviruses carrying the ADA gene. Now these lymphocytes are corrected with the normal ADA gene. Lymphocytes with normal ADA gene. Now these lymphocytes are reinfused back to patient's bone marrow, or introduction of these lymphocytes back to patient's bone marrow. Once these bone marrow cell gets the corrected lymphocytes, there in the bone marrow this gene, ADA gene, gets activated and starts to produce the normal ADA enzyme. Here in the bone marrow, contains corrected lymphocytes. So these corrected lymphocytes gets activated, gene gets activated and starts to produce the enzyme, that is ADA enzyme. So now patient is cured.